Okay, well, thank you very much for coming. This is the fourth of a series we have, Evening with the Genome Center, where we try and present topics of interest to the general community about the impacts of genomics on society. As I said, this is the fourth in the series, but they're very different, each one, and we're very happy to have suggestions of, if you think you'd like to hear more about a particular area of where genetics, genomics, maybe sort of high-throughput biology is of interest to you. Has anyone here actually been to any of the previous ones, or you all new? You have before. Which one were you at? You remember? Oh, you've been all of them. Great. Hey, here. See, you're returning. Okay, that's great. So tonight we're going to hear about viruses and sort of impact of large epidemics. I've actually known Dinesh for nearly 20 years. Scary thought. He was a postdoc in Barbara Baker's lab down in Berkeley, and he was one of the first people to clone a resistance gene in plants that allowed it to detect a virus and become resistant. So that was in the early 90s, very exciting time. And actually, there were 3 or 4 resistance genes that were cloned almost simultaneously. One against a virus by Dinesh, another against a bacterium. I can't remember what the third one was. Two against bacteria. So anyway, it was an exciting time in the early 90s. And then his postdoc, and then he moved to Yale, where he was assistant professor, associate professor. And then I persuaded him to move to Davis and the Genome Center in, when was it?
3.5 years.
What?
3.5 years.
3.5 years ago. Seems like a long time ago. Anyway, we're very pleased to have him here. And he's a real authority in plant molecular biology, viruses, and he's going to tell us some of his broader interests about the impact of virus in human health.
Thanks, Richard. Can you hear me? Actually, okay. So yeah, I know Richard when I was at Berkeley. Actually, so there used to be a common theme meeting, which we started again 3 years ago when I got back here. Actually, so thanks that you all here. I know it's Friday evening. So my broader interest. What we work on in the lab is to take both traditional approaches and the modern biological approaches to understand how organisms is able to cope with diseases, actually. Okay. So if you don't understand something, you can always stop me and ask me to clarify, and we can have as a discussion rather than just me talking all the time, actually. Okay. So I'm in the Genome Center. Is in the College of Biological Sciences at UC Davis. Here is the Genome Center building opposite to the football stadium that you see from away, actually. So if you have not been there, I recommend you visit sometime to see what's going on inside, actually. Okay. So next slide, please. So what I want to start with is, although want to bring back to the viruses which infects the humans, probably a couple of them I'm going to talk about today. To see how the modern biology is helping us to understand the viruses which could evolve as the pandemic viruses, actually. Okay. So before that, I want to give you some conceptual framework of where we all started, actually. So the concept of virus is only about 120 years old, actually. Okay. So next slide, please. So the scientist by name Bajornik, way back in 1898, coined the term actually virus. And what he was actually doing was he was actually a son of a tobacco farmer, and his father's field was infested with this symptom which is shown here, actually. So at the time, nobody knew what it is. What he was able to do is that he took some sap from the father's field and then started rubbing on healthy tobacco, actually. And then a few days later, the healthy tobacco got sick again, actually. So in fact, his father lost his farm because of the major infestation of this specific disease, actually. So he went down to identify what is causing that disease, actually. By the time we already knew that the bacteria exists, So he was able to eliminate other microbes from the tissue sample, and then using some of the microscopy followed by traditional approaches, he came to the conclusion that whenever you grind this leaf, a liquid, and you rub it, you get the symptom back again, actually. So later on, it turned out that it was named tobacco mosaic virus, simply because it makes this mosaic pattern of symptoms when the virus infects the leaves, actually. Okay. So believe it or not, the resistance gene I cloned, I was involved in cloning in Barbara Baker's group at Berkeley in 1990s, was the gene against this virus, which was identified as the first virus in 1898, actually. Okay. So later on, about 3 years later, the first human virus was discovered and identified to be yellow fever virus, actually. So if you go to the next slide. So if you, probably most of you are aware that the viruses are slightly different from other microbes, actually. Okay. So they are usually intracellular parasites because they need a living host in order to survive, actually, okay? In order to replicate, in order to multiply and spread, actually. So there is quite a bit of debate whether virus is a living thing or not. So we don't have to do that, actually. So for the conceptual framework, the viruses are usually intracellular, the parasites. And then also the genome could be either DNA or RNA, actually. So if you follow the central dogma in our own human cells, for example, so what happens is DNA, which have genes, and they get transcribed into RNA, and then RNA is converted into proteins called translation, actually. So most of the functions in the human's body is done by these proteins, actually. Okay. So the genes are there, but they have to be transcribed into RNA. RNA has to be translated into proteins, actually. And then the proteins, of course, will dictate a lot of other things like metabolism, everything. What happens in your human body is mediated by these proteins. In the last 10 years, of course, there is a lot of work is going on. RNA itself could have a function, actually. Okay. So prior to that, people always thought that it has to be proteins, actually. But there's a lot of work now that the cancer biology and all that stuff is partly driven by what is called as non-coding RNAs. So they don't make proteins, but they have function as the RNA molecule itself, actually. So, but compared to the human cells or other eukaryotic organisms like plants or flies, the viruses either contain DNA or RNA, actually. So they don't contain have a capacity to make DNA and RNA at the same time. So the infectious agent usually is a DNA virus or it's an RNA virus, actually. So once if it's a DNA virus, when it gets into the organisms, like in the human cells, it will convert into an RNA and then makes proteins and then take over the host cells, actually. So normally the viruses are also small. So here is a comparative sizing here. This is 1 micron, the smallest bacterium, and compared to the size here, the different viruses, we will talk about more on influenza virus today. So you can see the size of these are much smaller compared to the bacteria here, actually. So having said that, in the last 5 to 8 years now, big viruses have been discovered, actually. These are referred to as mini virus, actually. So these viruses actually could be as big as a bacteria, actually. So it's fascinating that these have been discovered more and more these days, and they in fact resembles having things both DNA and RNA. Prior to the discovery of mimi virus, people always thought it is either should be a DNA or an RNA virus, actually. Okay. So the next slide, please. So to put you in the basic framework here is that virus, in order to survive in a living host, it has to have achieved 4 basic things, actually. It needs to get into the host. Okay, we will talk a little bit about that. And then it should be able to, once it's inside the host, it should be able to make more virus, more copies of its genome, if it's a DNA or RNA. And then it should move systemically from host to host, actually. Okay? So the transmission, actually what makes the virus, whether it is really infectious or not, actually. So we will come back to that when we talk about influenza later on, actually. Okay? So what viruses have done is smart enough that they will have all the things they need in order to do these 4 things, actually. But of course, they will hijack rest of the proteins from the host cell, actually. Okay? So they don't leave anything for chance here, because they pretty much have everything they need to whatever they need to do to hijack the host to entry, to replicate, and then transmit from one individual to another individual. Next slide, please. So how many viruses on the earth? Is any guess? Millions. Next slide, please.
We including bacteria?
No, you could include bacteriophages. Yeah.
Bacteriophages.
Yes. Yeah.
The most
Numbers. Yes. So there are 10 to the 31 number of viruses on the earth, actually. Okay. So this is almost 100 million more than the stars in the total number of stars in the universe, actually. Okay. So the reason this number has been estimated is because people just beginning to find this large number of virus population in the ocean, actually, which people have not paid attention to only in the last few years, actually. So if you just take a liter of seawater, so there is 10 billion microbes like bacteria and archaea, but there are 100 billion viruses to be estimated, actually. Okay. So these viruses, in fact, infect these microbes in the sea. And this is a major force in terms of recycling the elements like nitrogen, phosphorus, and carbon, actually. So that's why people are, the scientists are estimating that they have a major role in planet's biosphere, actually. Okay. So there is a major interest now in trying to go and find what these viruses are. People only know very few. They've only isolated few so far, actually. So there are a vast number of viruses. We don't know what they are, actually. So if you have kids or grandkids ready to work on science, here is an interesting project to get into, actually, I think. And this also has a major impact because people speculating that some of the viruses they've already identified has major impact on biofuel, and things like that in terms of the fixation of elements and also recycling of elements in the biosphere, actually. Okay. Next slide, please. So what I'm going to tell you today is a couple of viruses because most of you probably maybe on the side vaguely follow these viruses, and it's also important in terms of the public health, actually. Okay. So the one is the influenza virus, which is the flu virus. So the flu season has just started, and this is the major virus which is responsible for causing flu, actually, influenza. So here is the electrogram of the virus, how it looks. If you look under this, you can see these spikes coming out of the viral outside. And if you look at the structure, which is depicted here, actually. Okay. So I know we don't have to go into the details of this, but few things you need to remember for the discussion today, actually. So the virus usually has these spikes outside, and there are two colors you can see. So one is HA, the other one is NA here. HA is for hemagglutinin, and NA is for neuroaminidase, actually. So the reason this virus actually can enter the host cell is because of the presence of this protein here, the spike. Okay. So that's what makes this virus to get entry, actually. Okay. This becomes important as we go along. And the reason the virus needs this protein also outside is because once it gets in, when it is coming out, because it is going in, it uses the host membrane. Coming out, if it doesn't cleave the membrane, all the virus is going to clump together, actually. So what this enzyme does is help to cleave off wherever the attachment of the virus and the host membrane, so the virus can leave and then infect another cell actually. Okay, so these are the two important components which we will be discussing today on in terms of the influenza here actually. Of course, there are other important things here. All the things you see inside here, there are 8 segments here. So these actually encode 8 different genes, and all these genes are required for virus to replicate in the cell and then package it and then get out of the cell and infect another cell. Okay, and we know quite a bit about this outside spikes, and this protein hemagglutinin or HA assembles into this cartoon structure like molecule, and this is where it binds to the host cell actually. Okay, so in the next slide, so what happens is that here is an influenza cartoon, and you have this HA-NA spikes, and it uses the HA to bind to the receptor on the human cell. So this is a cartoon of a human cell. Okay. So once it binds here, there are processes which makes this virus to get in actually. So once it gets in, all these 8 segments get disassembled, go into the nucleus, and starts making their RNA now actually. Once they make the RNA, they get translated into the protein, and they get packaged, and then they get out now actually, and then start infecting the another cell now actually. So here is where, in order to get out, you need this guy, the NA actually. If you don't have it, so then it gets stuck there. In fact, Tamiflu you take as a medicine is a target of the neuraminidase actually. So it basically inhibits the neuraminidase not to do its function of the exit here actually. Okay. Next slide, please. So the influenza is an interesting virus from public health because, as you probably know, they have multiple hosts. The most of the influenza infection is on birds or swine or the horses actually. So we'll come back to that, the impact of that on the humans actually. And every year, the seasonal influenza, which we all get occasionally every year, actually infects about several hundred million, and it has a mortality rate about 250 to 500,000 people globally actually. And in fact, go ahead.
Why is it seasonal?
I will come back to you on that. Oh. So the seasonal is because there are lots of changes and lots of reassortments happen between these different strains of influenza. And we know that some of the strains persist, and it is being persisted from long time actually, whereas some gets evolved newly actually. So at the end of the talk, I'll come back to you how actually the CDC, World Health Organization, and the rest of the world get together every year twice to decide which strain we need to make vaccine for the following year.
Okay. Why is it more important in the winter than the summer?
Because the virus has a tendency to go from the different places to move around actually, and then also the cold conditions favors the virus to be able to infect your upper esophagus, upper respiratory tract, or the lower respiratory tract actually. So there are again the different strains which is able to infect actually, and this is also something to do with immune response. So the children and the elderly are much more susceptible to even to the seasonal influenza compared to the young adults, like 20 to 40-year-old actually. But that is not the case in one of the famous virus. I will come back to that soon.
Okay. How does it differ in the tropical countries that don't have a cold season and the warm?
No, that's because there are different strains which can survive there actually. Yeah. So because it's all reorganization of those 8 segments I told you. I will show you soon what happens actually. So, yeah, next slide. Okay, so when you hear influenza, normally we talk about A actually. Okay, so that is always the problem one. Influenza virus A actually. Of course, the vaccine also includes B, but never C so far actually, because it's not a major problem actually. So most of the time I'm going to talk to you today is going to be influenza virus subtype A actually. Okay, next slide. So this is what happens here actually. So let's say you have virus A and virus B, okay, and both can actually infect human cell. So what happens is that they co-infect a single cell actually. So now you have 8-segment genome here, 8-segment genome here, but they are slightly different actually. So you can imagine now they can actually rearrange as soon as they are inside actually, because once they start replicating, 7 of these can bring one of these together, or 7 of these can take one of these together, or possible combinations are about 256 actually. Okay. So this is the major reason why there are so many strains of influenza, and which is why it is very hard to deal with influenza virus actually. And if you go into the next slide here, so another thing you need to remember is that there could be infection of 2 different viruses onto the same type of cell, like say human cell, but it also could be an infection on a bird and also a human actually. So that makes it much more complicated. As we go along, I will bring some discussion into that actually. So this is what is referred to as antigenic shift actually. The shift meaning there's a major genetic reassortment. So things are getting shuffled, and then new strain of influenza can be developed actually. Okay. So but there is also an antigenic drift, but that is very rarely noticed. So that is the mutations within the same genome actually. Okay. So normally what happens is the mixture rather than the mutation within the same genome actually. Okay. Next slide. So when you see or when you hear in the NPR or something, so normally they say, oh, there is a prevalent H1N1 strain actually. What does it mean? So what does it mean is that normally you name the influenza strain based on what is the type of influenza. This is type A. Where was it first isolated? In this case, Singapore. What's the number of isolates they isolated? Specific to this one, there were 6. Here it was isolated in 86, and the major type change is in these 2 proteins. Remember, HA is required to get in, NA is required to get out actually. So a lot of the changes you see always in these 2 proteins. It makes sense, because in order to get into a new type of cell, HA has to change actually, and in order to get out of there, NA has to also change actually. Okay. So there are more flu strain evolve in Asia simply because of the vast number of the poultry and wild animals which are close to the domesticated animals and also people actually, okay, compared to the other places actually. So we will come back and talk about a couple of strains actually. Next slide please. Okay, so this is a complicated slide, but I want to take home few things here. One is, if you take a century, 100 years, okay, every 30 or 40 years there is a pandemic actually. Okay, so it's been recorded since 1700. Every 30 years there is a totally new strain comes along, and it infects majority of the people and causes a havoc actually. So the classic one, which always people talk about, is this 1918 Spanish flu. Okay, so 1918 Spanish flu is still talked about it like a plague, simply because it killed almost 50 million people worldwide actually. Okay. So India alone, there was 18 million deaths in India out of that 50 million actually. Okay. So, but if you go back now, and I'll come back to that, how scientists are trying to understand this using genomics and modern biology tools, is that it is hard to trace how this strain evolved actually. Okay. So the one theory is that there were 2 different strains. One was from the avian strain. The other one was the mammalian strain, where it can infect the human and the pig. And somehow they 2 got together and then evolved into a new strain. You can see the color here is changing. So that means it reasserted actually. And then eventually it became so infectious onto the human that it became a pandemic. Okay. So similarly, if you take here now, so H1N1 was controlled until like 1957 actually. But all of a sudden there was some change happened again actually. So H1N1 combined with an avian strain H2N2, and then H2N2 reasserted and became infectious heavily on humans again actually. So there was another small pandemic here, not compared to what happened in 1918 actually. And then you can see the H2N2 combined with another avian strain here, and in 1968 in Hong Kong, this was a major problem again. So this is an interesting one. Normally what you see is two strains getting reasserted to create a new strain actually. But here there are three different strains got reasserted here actually. So one was from the seasonal flu, the other one is from the avian, and the other one was from swine actually, the pig strain. So you can see this one eventually became heavily infectious on human. This is actually the one which started in Mexico and the United States and spread very quickly in 2009, almost 30 countries actually. Okay. So you can see here these kind of genetic reassertments is what makes these viruses to evolve, because normally they are in the swine or in the horse or in the aquatic birds, but they change something by genetic reassertment to create a strain which is going to be much more deadly on people. Okay. Next slide. So what do we know so far? So there are 15 types of H, okay, and there are 9 types of N actually. So you can figure out the combinations here. So we know all this because by genome sequencing, all this stuff. I will come back to that soon actually. But right now, the World Health Organization and CDC is much worried about a strain which just came along in May of 2013. This is the H7N9 actually. So far, there was no recorded infection of H7N9 on human actually. Okay. And it already had 130 cases within the span of 5 to 6 months. And there are 40 deaths actually. So this is almost like 20% mortality so far. Okay. So I will tell you now, H7N9, the dramatic work which has happened in the last 6 months using the genome sequencing information to figure out where is this virus came from actually. Okay. So go back, next slide please. So what they did, the scientists did, was they conducted a surveillance from April 4th to 18th, and then they went to these different provinces, 500 kilometers south of Shanghai, 600 kilometers north of Shanghai, because this is the region where there was a major outbreak actually. And they collected about 1,300 samples from chicken, ducks, geese, pigeons, and quail. They also collected about 1,000 samples from the poultry markets, farms, wastelands, and things like that. And then, of course, the sequencing this will not tell you anything because you don't know what it is actually. Okay? So what is interesting about influenza is that there is a large collection of archival samples actually. So scientists have been collecting and archiving these samples. In this case, they used a collection between 2000 and 2013 from southern China actually. Okay. Most of these strains were collected from southern China actually. So what they did was they massively sequenced all these samples actually, and then asked what happened here in comparison to the archival samples here actually. So in order to figure out how this virus has evolved as a major outbreak virus just recently actually. The next slide please. So here is what they figured out. Okay, so I don't want to give you all the things which has gone on in the back burner because this is a lot of work. But remember, this is only within the span of 5 months. They were able to collect all the samples, genome sequence, the whole thing, and then figured out where this virus has come from actually. So you can see here is that this is the virus we are talking about, H7N9. So you can see in the uppermost here, these are individual strains of the viruses, how they actually recombine and reassorted, leading into this H7N9 actually. And what is interesting is that if you follow these strains have never been able to infect humans before actually. So only reason this can now is because of this major genetic reassortment in the genome actually. But what is also interesting is that they discovered a new virus, this H7N7, which is there from 2000 in the bird population, but people didn't know about it because there was no genome sequence information actually. So of course the relationship between this strain and this strain is not known, and I think this is a major serious threat currently. In fact, a couple of weeks ago in St. Louis, there was a meeting, World Health Organization and all the scientists, to discuss how to tackle H7N9 and how to start designing vaccine to this strain of H7N9 actually. So you can see a simple reassortment leading into a strain which was not existed before actually. So next one. So the same thing people were able to do using the genomic sequencing, high throughput sequencing. 2009, as I said, H1N1 was started in Mexico, and then in United States. Within the span of 2 years, it entered 30 different countries. So people even still think that this could be the next pandemic of 21st century. So this is really interesting one because the genome sequence helped to formulate what has actually happened. Okay? So this is a combination of 3 different strains of viruses from 3 different organisms actually. So one is coming from avian H1N1, another one is the standard seasonal flu virus from human, and one is from the swine population here. So if you follow the trajectory here, so there are 8 genome segments shown here. So until 1979, everybody stayed on their own actually. So all of a sudden, somewhere here in 92, there was a mixture between swine, avian, and the human virus actually. And by 2009, this virus evolved as a new virus, which now can infect the human cells actually. And what is also interesting here is that because of the availability of the samples here, we can go back and ask when this virus really started. It's actually started almost 10 years before there was an outbreak actually. Okay. So why I'm saying you all this is because the genome sequencing is really helping us to go back in time and ask questions. Why did this virus really evolve actually? And this will have a major impact as we go along in the future. I will come back to that later. So I showed you 2 viruses, H7N9, which just got in, and the 2009 H1N1, how the genomics has helped to sort out where these viruses came actually. Next slide, please. So I just want to give a plug here for UC Davis Genome Center. So the director you heard from Richard Michelmore. Assistant director is our David Siegel. He's back there recording this talk now. So we have a number of core technologies. So Richard was great in terms of envisioning the future of genomics technology in UC Davis. And from the stories I hear, after a lot of struggle, he started the Genome Center through UC Davis. And there are 5 or 6 platforms going from sequencing to proteomics to bioinformatics to whatever you name actually. Okay. So the DNA core technology has these the sequencing equipments, which is what people are using these days to sequence the genomes that I just talked to you about actually. And each one has its own advantages actually. So for example, this one, MySeq, can actually sequence one of your genome within maybe 2 days. But of course, the analysis takes longer time. And this one gets a lot more sequence, but it actually takes longer to run actually. And then this one, for example, has more advantages compared to here. These guys only give you small reads of 50 nucleotides. but this one will give you a long read of almost 1,000 nucleotide actually. So the reason I'm bringing up here is that it is also important to foresee where the technology is actually. So in that sense, Richard always knows what's the next technology that we should try out at least actually. So this happens to be one of the 3 or 4 units which was delivered to UC Davis to test it out when they released it about a couple of years ago actually. And of course there is a technology coming called nanopore. You can probably sequence your genome for $1,000. That's what the claim is. And also you can even put the blood into the USB and plug it into your computer, and a day later you should have your sequence actually. But of course, you need somebody to analyze all that actually. Okay, so these are the tools in the genomics which is helping to do some of the things I've been talking about so far. And if you want to look at them, there will be sometimes open houses that you can stop by at the Genome Center actually. So next slide, please. So I showed you the most recent 2 viruses which how genome sequences helped actually. So in this area, especially influenza, since every 30-40 years there is an outbreak, the scientists always want to go back to understand what really happened during those times actually. For example, 1918 flu virus. Why did it was so devastating? Why did it kill 50,000 people actually? So knowing that information will help us to foresee what could be coming actually. Okay. So I'll give you a couple of examples here actually. So 1918 Spanish flu actually killed more people than the World War I during that time actually. So these are some of the cartoons. This is in fact a Stanford hospital tents very close to us from here, an image taken from this virus Stanford. So what people trying to do is to ask, why was this so devastating on the human population actually? Next slide please. So here is again another technology which was very useful actually. So there were some autopsy samples and also some frozen lung tissue samples actually way back from the patients, almost 1918, 1920. So it's 100 years ago almost. Okay. So the technology is so advanced that before you have to have a lot of DNA or RNA in order to do sequencing and all that stuff. So now you don't need actually. You need very little DNA and RNA in order to go in and find out what is there actually. So using these samples, people were able to sequence the 1918 strain actually. Okay. So next slide. So what was interesting is that when they sequenced the strain, it didn't actually give any clue. Why was it so aggressive? Because there were a few changes, but other than that, it's almost same as the existing strains actually. Okay. So here is another technology. It's not genomics, but it is derived from genomics, is what is called reverse genetics actually. So what was there in 1918, and then you do a reverse genetics to recreate that actually. Okay. So you can create all the 8 segments which were there in 1918 actually, and you put it back into the host cell and ask, does it now infect as aggressively as it infected in 1918 actually. So by doing so, they recreated the virus here actually. Okay. So still it didn't give them any clue actually. So the one clue which came along was, next slide please. So only few changes was required to change from infectious to non-infectious actually. Okay. So just making a couple of changes, the transmission of this virus was completely eliminated in the human cells actually. But of course they use a model for these called these are ferrets, these are the polecats, because this has to be in the mammalian system in order to make any conclusive conclusions. So what is shown here is that the human influenza virus using this HA binds to this specific receptor in the cell. This receptor is outside the cell, virus binds and gets in. It's through this HA protein we have been talking about actually. Okay. But in the case of the birds, it's a different kind of a receptor, almost similar, but it is different actually. Okay. So the human one cannot bind to this receptor, but the bird one cannot bind to this receptor. Actually. So what they figured out from the flu virus is that just making 2 changes, you can now make that virus infect bird, but not human actually. So you can imagine just 2 amino acid changes in this protein here actually. So that gave them the clue that there was some subtle change happen. That's the reason it had a major problem. Actually. Okay. So next. But can we generalize that? Is it a generalized phenomenon that occurs in these kind of cases? So a classic example they took to address this is this H5N1 actually. This is also a pandemic influenza which started in Hong Kong actually. Okay. So the reason they took, next slide please, is because H5N1 was not as deadly as it should have been because it was not transmittable actually. It infected people, but from aerosol it was not going into the other people, which is a good thing because otherwise it would have been deadly actually. So why? Why was it not able to transmit actually? So the reason is this. So the bird one will bind to this receptor, the human one will bind to this receptor. So this receptor is present in the upper respiratory tract cells actually. Okay? But the humans also have this receptor, but this receptor is present in the lower respiratory tract. So the virus H5N1 can go in and stay in the lower respiratory tract, but it cannot come out through the aerosol because it is long distance to come out actually. So all these studies suggested that the few changes you can make, you can actually change the way the virus behaves in the human population actually. So of course the question is, next slide please, why it cannot be transmitted? Can we change it to do so actually, so that it will give us an idea in the future what makes these viruses deadly transmittable viruses instead of just infecting a local population actually. So can it easily evolve into a transmissible strain actually? Okay. So here again the genomics came into the picture because they mutated this specific HA sequence to almost 2 million combinations actually. Okay. So of course in order to know those combinations you have to go back and sequence all of them. Okay. So as soon as they sequenced the one which was able to bind to the receptor in the upper respiratory tract kind of receptor, they figured out there was only 2 amino acid change in this HA protein again, which is required to bind to the human receptor actually. So what they did now is that they took this mutant and they put it with the H1N1, 7 genes. This is 1 gene here, and they put it on the ferrets. Within 1 week, they were able to recover another mutation actually. Okay. So 2 became 3. And then when you take the virus from that and then infect the ferrets, now it is easily transmittable by aerosol actually. So next slide please. So interestingly, all you needed was 4 changes. That's not a big deal for influenza, okay, especially when there is so much recombination, reassortment is happening actually. So you can imagine, although H5N1 is now not transmittable, but there is a possibility that it can actually mutate to be transmittable strain actually. Okay. Next slide please. So these are some of the recent work just few months ago which came out to demonstrate these concepts, that how these so-called pandemic viruses have evolved and how they change in order to infect a lot more than what we expect them to do actually. Next slide please. So another question the scientists are interested in is in this 1918 flu actually. So in the beginning of the talk I said most of the seasonal flu is usually young children or the over 60 years old actually. But this one took majority of the people who were actually 20 to 40 years old actually, the younger population actually. So the scientists have been wondering why did it infect young people actually. So again here one of the genomics approach they took, this is actually quite old now even though this work is only 5 years old, But now we have a brand new technology in the last few years, which has revolutionized this technology actually. But since they use this technology, I'm going to tell you this technology actually. So this was there in the Genome Center, and now it is replaced by a new technology actually. So what you do here is that this is like a small glass slide, micron side glass slide, okay? And then you actually spot in individual RNA from the human cell actually. So you can synthesize those things, okay? So let's say the human cell has about 30,000 genes actually. So we can synthesize this 30,000 RNA complement to that gene actually. So you can put them on there, okay? And then you take the sample from the uninfected cell, and you take the sample from the infected cell. Uninfected cell RNA you label with red color, we can do that. And the infected one you label with green color actually. So you mix them together now, and you put it on this chip here, which has all the genes in the human cell actually, RNA, okay? So now if any of these RNAs bind to these RNAs, they will light up actually. And that's what you see here actually. So the green means it is majoritarily bound by the infected sample. The red means it's majoritarily bound by the RNA from the non-infected cell. Mixture means both can actually bind to the RNA actually, okay? So the output is, next slide please. So the output is if the genes are induced, meaning going up in their expression, they all will be red actually. If the specific genes are going down in their expression, that means they are all going to be down actually. So using this, they were able to figure out why 1918 virus was so influential on the younger adults, not on the other sectors actually. Next slide please. So here is why. So what they did here is that they did the classic microarray experiment I just told you, okay? So they took this strain of the virus. This is a Texas strain, which is a very mild influenza strain, which the people use it as a control. And then here is our reconstructed 1918 strain actually, okay? And these ones have part of the 1918, but not full of 1918 actually, okay? So you can see there are 3 different days here after the infection of these 4 different viruses. And these are different cells. So these are the cells why we are alive, because these are the cells which take care of the fighting against the infection actually. The neutrophils, macrophages, natural killer cells, especially for virus, and the Th1 cells. So they all produce cytokines and interferons, which is able to fight against the virus infection by recognizing the virus which is coming in actually, okay? So if you look at the mouse lung infected with this influenza 1918 strain here, you can see there is a lot more red in that lane compared to the Texas strain, which is a mild strain actually, okay? But also in addition, it is persisting from day 1 to 3 to 5 actually. It's not going away. Whatever was up, it's staying up actually. Next slide please. And also if you look at this one here, a similar kind of experiment, which a control strain and a 1918 strain here. So the day 3, in fact, the control strain is inducing a lot of these genes are induced by interferon, which is what you need to fight the pathogen actually, because this is an innate immune response which is activated as soon as the pathogen infected actually. So in day 3, they both look same actually. But if you go to day 6, the genes are still on in the 1918 strain, and also day 8, but the genes have come down already in K173. Okay. So the reason why many of the, next slide please, the younger adult was infected is because their immune response, they are able to activate immunity much higher level than the younger kids or the older people actually. So when you have so much of immune response activated, it becomes an autoimmunity actually. So you are overdriving the immune response actually, because you want to activate to stop the virus, but not to activate continuously, which leads into a tissue damage actually. So once happens, then these immune cells is going to take over where they should not be there to begin with, actually. So you can see a simple experiment when they did to identify what happened, they couldn't figure out because the sequence looked almost same. But doing this kind of high throughput expression analysis, they were able to figure out the reason was the immune cells are hyperactive in terms of activating immune response, actually. Okay. Next slide, please. So new influenza strains appears each year, as we discussed by reassortment. Here is a Google global flu trend, just 5 o'clock. So Google has this map that you can follow what's happening with the flu throughout the world, actually. So right now you can see it's mild in most of the places, actually. But if you come back by January or February, I think the color will change to red in the United States, actually. So the next slide, please. So here is a map yesterday, actually, of the US. There are some infections already in the regional states. California, there is no major issues so far. The color says how infectious is how widespread is the flu right now, actually. Of course, it's still November. You don't see much of a color in the US map here, actually. So what happens each year is that because of this major influence on the different strains, there is a constant surveillance, actually, in the United States. Every week you can watch a graph, what's happening throughout US, actually. So there are about the CDC and also the World Health Organization. So there are 130 US cities which have been monitored for the influenza illness or the cause of death if it is due to influenza, actually. And of course, this is coordinated with the worldwide effort. So there are about 101 countries participate in this sharing of the data of the surveillance, what's happening with the flu strains. Next slide, please. So of course, the prevention and treatment, so all the things I told you so far is that the genomics, I think, will help us to do this much faster than what we can do right now, actually. Okay, because the surveillance is the most important part, and collecting samples and knowing what kind of strains are there will be helpful to design vaccines and things like that. And so far, there are only 6 of the 15 influenza A subtypes. As we discussed, the new one is the H7N9, okay, which should be put, the human picture has to be put in here. The next slide, please. So for example, in the case of H5N1 in Hong Kong, when it came along, you can see a number of agencies, the CDC, United States Department of Agriculture, Food and Drug Administration, National Institute of Health, all these agencies came together and did a surveillance immediately to figure out what is this H5N1A, and also have a seed virus so that they could start the vaccine immediately, actually. Okay. So the next slide, please. So what is important is, I think the scientific community knows now, is that the surveillance not only in the people, but also surveillance in terms of monitoring the other populations, actually, the swine and the horse and the aquatic birds, the chicken, poultry, all this has to be surveyed, actually. Okay. So the genome sequencing effort, the capacity to do genome sequencing actually helps these processes now, because we can sequence relatively cheap, and also we can do it much faster than what we could do before, actually. And these are going to be very important in terms of designing the future vaccines to avoid any pandemic which can happen due to genetic reassortments, actually. Next slide, please. So the influenza vaccine, so we all go and take the vaccine. Just want to give you an idea here. So the antigen is decided in February of each year for Northern Hemisphere, and September for Southern Hemisphere. So the committee, which includes US CDC, World Health Organization, and many countries, meet twice a year, and they decide which strains to be used to make the vaccine, actually, because it almost takes about 6 months to make the vaccine before it can be delivered to the people, actually. Okay. So right now, this year, there are two vaccines. One is a trivalent vaccine, which is influenza A virus, H1N1, H3N2 strain, and one influenza B actually. But there is also a quadrivalent vaccine which has been produced, which includes the second type of influenza B, which seems to have some emergence in the last year actually. So you can see which vaccine to make depends upon the surveillance actually. So better surveillance you have, better predictions you can make, better prediction of mathematical models you can make, better will be the vaccine against when you administer it. Okay. So of course it takes about 2 weeks once it is given, because your immune cells has to produce the antibodies against the vaccinated virus before it can actually be effective. And even though somebody can say, oh, I have taken it previous year, you still have to take it, because the strains could be different. And also the immunogenicity actually comes down as the year goes along actually. Okay, so next slide please. So get your flu shot. It's probably available now. Next slide please. So I think, of course, if you look into all the things about influenza, we have to be very careful. We have to be diligent, and we have to do a lot of surveillance and do the work which is going on in order to avoid the next pandemic actually, because this is a major public health threat because of the way the virus can evolve, and also has the capacity to infect a wide range of geographic area actually. And also one thing to remember is that in 1918, not many people moved from one place to other. So these days, in a day, so many people go across the borders actually. It's not anymore one country, one virus. It's global virus, and your local virus could be same as in China or in Davis actually. Okay. So next slide. So I have only few slides here. So the second important virus I just want to briefly touch in the next 2-3 slides is in 2003 this virus came along actually. This actually made a major issue in 2003, if some of you remember actually. So this originated in actually in a hospital very close to a building in Hong Kong. This is because it originated in a small shop there, and then it infected the health workers, and then it got transmitted to other people because of the close contact. So the good thing about this virus is that it does not transmit through aerosol. It has to have a contact based actually. So the next slide please. So what happened basically is there was a direct contact with the healthcare workers, and then they contacted some of their family members, and of course then it got out of the Hong Kong, and it spread into different places actually. So the next slide please. So what is also interesting here is that it took almost 7 months here before they could figure out what virus was it actually. Okay, so we are talking 10 years ago technology now. So if it were to be now, in a week you could probably figure out what virus was it at least actually. So they figured out this was a new coronavirus by using a lot of different technologies, and then they also demonstrated that the virus is transmittable by infecting this the cat. And eventually the vaccine was also tested against the same kind of animal actually. Next slide please. So you can see in 2003 there were almost 9,000 cases of infection and 900 deaths starting from Hong Kong throughout different parts of the world actually. So why I'm telling you is this. So this is the 10th anniversary of the SARS discovery of outbreak actually. Okay, next slide please. So now we have SARS to MERS actually. So just few months ago, in April, there was a new strain of the Middle Eastern Respiratory Syndrome strain was discovered in Saudi Arabia actually. So there's already a 50% mortality rate here actually within the span of 6 months. 114 confirmed cases, 54 deaths actually. And the virus has already spread from Saudi Arabia into France, Germany, Italy, Tunisia, and United Kingdom actually. So I think we have to be careful in terms of surveillance, and so that it helps to contain the virus, especially in the SARS. The containment is much easier than the influenza actually. So the next slide please. So very nice work just 2 weeks ago came out of part of the scientists in this work is from UC Davis. So what they've been suspecting is that the coronavirus can actually live in the bat population actually, okay, as a reservoir. And then eventually, once in a while, it can actually jump into humans actually. So what they did was they used the genome sequencing to figure out what this coronavirus in the bat, and they reconstructed the virus. Interestingly, it can actually infect the humans using the receptors which is already present in humans. Okay. So it doesn't need anything else. So the surveillance of this population is very critical in order to monitor what is happening with this coronavirus, because it already has a receptor which is amenable by the human cells. Okay. So next. Okay. So thanks for coming. Hope you enjoyed. So you can see a lot of the modern technologies helping us to dissect these things to understand what is happening. And at a bigger scale, I think, is the World Health Organization and CDC has been very good in monitoring the surveillance to design vaccines. Of course, there is a lot of work I didn't talk to you about from NIH, National Institute of Health, on the vaccine development itself, actually. So for now, the vaccine development is slow. It takes about 6 months, okay, because we still use the traditional way of making vaccine, actually. But there are a lot of new technology people are testing right now, something called DNA vaccine or RNA vaccine. That could be delivered, if it works, within a span of 2 to 4 weeks, actually. So that is what you need when there is a pandemic. You cannot wait for isolating the seed virus and start the seed virus to make the vaccine, and then you have to test it before you actually deliver it. Okay. Thanks.